Hello students, today we are discussing a topic which is very important in life sciences, genetics. Genetics, generally this is a branch of biology which is born in 90s after a rediscovery of Mendel's work. Genetics means that the science which deals with the heredity and variations amongst the living organism which we study during study of life sciences. That Mendel we call as the father of genetics. Before Mendel there is no such branch of genetics, but Mendel first told us that how the character inherited from one generation to another generation amongst plant, as well as he also concluded that such inheritance is also possible in case of animals. Mendel in 1860 conducted a pea plant experiment. A pea plant scientifically known as a Pisum sativum. Mendel firstly collected a lot of varieties of peas, but he selected only Pisum sativum because Pisum sativum is such a variety which having both abilities, having self-pollination as well as cross-pollination. Due to the self-pollination, Pisum sativum is a true breeding variety, and such true breeding varieties means homozygous varieties for number of characters can be easily handled for hybridization. So Mendel selected Pisum sativum. Mendel did his famous pea plant experiment in 1860s. Mendel's work on pea plant published in 1866. Mendel considered 7 contrasting characters of Pisum sativum. It includes the height in plant, it includes the flower color, position of the flower, pod shape, pod color, seed shape, seed color. So concept of Mendel is like that. He firstly tried to do the monohybrid process. So in the monohybrid process, what is monohybrid cross? Monohybrid cross means when we take the two homozygous parental varieties, and these two homozygous parental varieties having two contrasting forms of a single character. Example, if height in plant or height is considered for character in pea plant, so Mendel selected two such homozygous plant, one parent having a tall height and another parent having a dwarf height. Both parents are homozygous, and after crossing these two homozygous parent, F1 progenies all are hybrid, all are heterozygote. So, but after crossing, when all F1 progenies all are heterozygous but shows only one character which is a tall in height, another parental character is not appeared in F1 generation. So Mendel think that how this character only one parental how this character appeared, and is it any sex influences on the inheritance of character? So Mendel did the reciprocal cross to verify that if there is a influence of sex, male sex or female sex on inheritance of character, and after the reciprocal cross also same result appeared, all the F1 progenies having only tall character. So Mendel concluded that tall character, which is one of the parental character, is dominant one and another character may be recessive. So Mendel tried to verify that what happened when we self all the F1 progeny amongst themselves to get the F2 generation, and after the selfing F1 progeny amongst themselves, Mendel got the F2 progenies, and F2 progenies having very interesting result. Why? Because the character which is not appeared in the F1 generation again reappear in F2 generation. Definitely its a percentage or its a ratio is very less. It is 1/4, okay, or 25%. But definitely this character reappear in F2 generation. So Mendel concluded that the character which is unable to appear in F1 generation because 2 units of heredities are there. So in Mendel's time there is no concept of gene. So Mendel called it as a factor. So 2 units or 2 factors are there, okay, for each form of character or trait, and when the dominant form present with the recessive form, only dominant form appear and recessive form unable to appear. But during the gametogenesis, when segregation of the dominant form from the recessive form happen, so after homozygous condition of the recessive form again appears in F2 generation, and we call famously as a principle of segregation or law of purity of gametes, okay, which is firstly given by the Mendel, Now you know that the famous monohybrid Mendelian ratio is 3:1. Okay, this ratio belonging to the F2 generation of the monohybrid cross. And after did this successful monohybrid cross, and after getting the successful 3:1 ratio, Mendel can also consider that what happened when we consider 2 characters at a single time and how these characters inherit, okay, in future. So Mendel considered 2 characters means 2 different genes and their controlling character, and Mendel tried to study the inheritance of these 2 characters. So in that case, Mendel did dihybrid cross. So in that case, what happened? What is dihybrid cross? Dihybrid cross means when 2 homozygous parents crossed for 2 pairs of contrasting characters in F1 generation, definitely all our dominant character appeared, but in F2 generation ratio is different. So Mendel also concluded with this experiment that 2 kinds of progenies appeared in F2 generation: parental types and new combination. We call it the recombinant. So you know that principle of independent assortment arises, means this is second principle, okay, of the Mendel study. So in this case, what happened? Suppose example, Mendel considered one parent which is homozygous for yellow seed color and round seed shape, another parent having green seed color and wrinkled seed shape. Such both the parent homozygous for both the character, homozygous means pure line for both the character, when crossed all the F1 progenies are hybrid one, we call it the dihybrid one. And then again same process repeated, all the F1 progenies selfed amongst themselves, and after selfing or crossing amongst themselves, Mendel got 16 progenies. Out of 16 progenies, 3/4 progenies, okay, shows a certain different characters. Especially 4 kinds of progenies arises in the F2 generation: yellow round, then next type is a yellow wrinkled, next one is a green round, and last one is a green wrinkled. So dihybrid ratio arises after the selfing of dihybrids, F1 hybrids, 9:2, 3:2, 3:1. So 9:2, 3:2, 3:1 is a famous Mendelian dihybrid ratio. Now you think about that, this ratio is a very significant. Why? Because 9 plants shows parental phenotype, yellow round. 1 parent shows parental phenotype, which is a recessive one, and 3 means 6 progeny shows the new combinations like a yellow wrinkled and green round, which is not appeared in any parental character or combination. So Mendel concluded that the genes those control different characters do not dependent on each other for their inheritance. Means every gene is independent for their inheritance. So during the process of inheritance, both gene get segregated, and may after segregation both creates new combination, and some creates parental combinations. Now you think about that, when law of independent assortment or principle of independent assortment we try to state, we can state that when two homozygous parents crossed for two or more pairs of contrasting character, inheritance of one character is independent from inheritance of another character. Means you see that this is for only dihybrid cross, and it's like inheritance. It's like a inheritance. This is a typical Mendelian experiment. So monohybrid cross and dihybrid cross, these are the basic crosses did by the Mendel, and after these crosses Mendel got two principles, law of segregation and law of independent assortment, and these two principles gives direction to the future genetics discoveries, because people know that some factor or genes are there, those are responsible for the inheritance of character from one generation to the next generation. And if we call it the units of heredity, so in case of plant as well as animals as well as microorganism, every character controlled by the segment of DNA we call as a gene. Segment of DNA we call as a gene. Now one more aspect is there in genetics. With the heredity, variation is also another aspect. Means why a single gene, its character, its phenotype having variations, and this is only due to the mutation. You know that any gene in the individual organism, okay, do not as it is remains for longer time. So there is a certain natural mutation or artificial mutation happens in any living organism. It may be in bacteria, it may be in case of animal, it may be in case of the plants, and after that the wild type character get changed with certain variations, various phenotypes we call as the phenotypic forms. So definitely what happened, a single character, single trait having more than one phenotype, and this is only due to the mutation. This is only due to the mutation. So mutation is a change in gene, okay, and it may happen naturally or artificial. It may happen naturally or artificially. It means that after Mendel, Morgan is another scientist who studied mutation, okay, means natural mutation as well as artificial mutation, and genetic progress turn into the discovery or much more research in the molecular biology. Thank you.